Now we'll see about the very short answers, that is 2 marks answers. We can't say particularly that actually this answer may come in the examination, but many 2 marks answers are there. But important we are discussing here. If you are starting from the enzymes lesson, in the enzymes particularly 4 digits in the enzyme code. The 4 digits in the enzyme code mainly indicates what? The first digit always indicate about the major class, second digit always indicate about the subclass, then third digit always indicate about the subclass, and fourth digit always indicate about the serial number. Then in the enzymes only, if you are discussing about the cofactor and apoenzyme. Apoenzyme is a protein part of enzyme. Cofactor is a non-protein part of enzyme. Apoenzyme to continue its function compulsory it requires the cofactor. The cofactor always participate in the enzymatic activities. Then in the enzyme activity only particularly, enzyme action has been described by the different mechanisms. In that mechanisms particularly lock and key model we had, and then induced fit theory we had. Lock and key model has been proposed by the Emil Fischer. Then induced fit theory has been proposed by the Kochland. Right? This is about the enzymes particularly. These are the important points in the enzymes we are saying. Apart from the enzymes, if you are taking particularly in the plant growth and hormones, particularly in the plant growth and hormones, quiescence we had, and then dormancy we had. Quiescence it means what, and dormancy it means what? Quiescence particularly it means it is a phenomena in which seed dormancy occurs always by the external conditions. Dormancy is a phenomena in which the seed is not germinating particularly by the internal factors, sometimes also by the external factors. And apart from this thing, in this plant growth and hormones, apical dominance we are saying. Apical dominance is a phenomena in which what is happening? Apical bird is suppressing the growth of the lateral bird. Apical dominance is actually promoted by the auxins. And bolting. Bolting is a phenomena of the sudden increase of the internodal length. It is promoted by the gibberellins. Then Richmond-Lang effect. Richmond-Lang effect particularly is by the cytokinins. It is always a phenomena of the delay of senescence of leaf. Whenever the nutrient absorption is taken more, nutrient secretion is taken more, then the delay of senescence of leaf takes place. That is the Richmond-Lang effect we are saying. And from the photosynthesis and respiration particularly, the common points which we are seeing, the C3 and C4, and then about the different enzymes and all these things, substrate level phosphorylation, first substrate level, second substrate level, and then about the respiratory quotient. Respiratory quotient is a phenomena always we know actually number of carbon dioxide molecules evolved by number of oxygen molecules consumed. That is also important for the 2 marks. Then very important things, that means actually C3 plants, C4 plants, acceptors, carbon dioxide acceptors, and all these things we have to see in the photosynthesis and respiration, all these things. Then if you are moving for the genetics particularly, in the genetics the chromosomal theory of inheritance has been proposed by the Sutton and Boveri. Test cross, the test cross is a phenomena of crossing between the F1 generation and then residue plant. And then about the phenomena of the point mutation. The point mutation, it means the base pair mutation which takes place we are calling as a point mutation particularly, important in the genetics. And then about the monohybrid cross-root, dihybrid cross-root, that means single parent is involved and 2 traits or a single trait is involved. All these things are important in the genetics phenomena particularly. And then about the, if you are moving for the molecular biology, in the molecular biology particularly if you are seeing genetic code, that means actually initiator codon, terminator codon. Initiator codon is always AUG, and terminator codons are always UAA, UGA, UAG. Then in the same molecular biology, then we are seeing about the capping and tailing. Capping is a phenomena which is always taken place at the 5' end. Methyl guanine is added, and the tailing is a phenomena which is added at 3' end, that is a poly A tail. That means adenine residues are added in the 3', that is a capping and tailing phenomena we are saying, that are important in the particular molecular biology. Then lactoperon concept, and lactosoperon has been proposed by the Jacobin Monod. And then in the particular things of the biotechnology, exon, intron. Exon is a coding sequence, intron is a non-coding sequence. Then the molecular scissors, restriction enzymes are the molecular scissors, and the cloning vector particularly. Cloning vector, what are the different sites we had in the cloning vector, about the marker genes, and then artificial markers. then selectable markers, all these things we have seen the cloning vector. Artificially plasmids, artificially designed plasmids, if you are seeing PBR 322 and PUC 119. PBR 322 particularly, if you add particularly, it is polymer plasmid, Bolivar rotary gauge. PUC it means plasmid of University of California, that you have to see particularly. Then biotechnology definition. Biotechnology is a branch of science which always deals with the production of the enzymes and new products with the help of the particular techniques involved in the field of the science. That is the biotechnology we are saying. And PCR technique, polymerase chain reaction, is very, very important. That is actually from one copy we are making the millions of the copy in the same time, in less time. That is the PCR technique. Denaturation, annealing, we have to indicate in the PCR technique particularly. And then different things we have seen in the biotechnology. And then downstream processing. Downstream processing is a phenomena of last step of the production of the product, that is the package, and then separation and packaging is done in the downstream processing, that we are seeing the biotechnology. Then in the particular thing about the last microbes in human welfare. In the microbes in human welfare, we are seeing particular actually immunosuppressive agent, particularly cyclosporin A is the immunosuppressive agent we are seeing. Then about the statin production. Statin production is actually done by the Monosmus purpurea. It is producing the statin production. Actually, Monosmus purpurea is a fungi which always what it is doing, it means it is not producing the enzyme which is producing the cholesterol. Then Swiss cheese. Swiss cheese is actually it is consisting of many holes, and the Swiss cheese is evolving the more amount of carbon dioxide. Swiss cheese is always produced by the bacteria known as Propionibacterium shermani, and nucleopolyhedrosis virus, which we had particularly, it is a biocontrol agent. Nucleopolyhedrosis virus, which we had particularly, it is attacking on the arthropods and all these things. And biogas production, and biogas which we had particularly methane and all these things we had important. Then GEAC, genetically engineering approval committee, GEAC particularly which we had. GEAC is always pointed to see the whatever developments which have been taken in the gene production of the gene, and then gene sequencing, biotechnology, RDNA technology, whatever which has been happened, it had been controlled by the genetically engineering approval committee. And green revolution we are seeing particularly. Green revolution, it means it is a phenomena of enhancement of the crop production. The father of green revolution in the world is particularly Norman Borlaug. The father of green revolution in India is MS Swaminathan. And gene revolution is a phenomena particularly. Gene revolution, it means it is a phenomena in which genes are applied for the particular increase in the crop production and formation of the transgenic plants, and all these things we are seeing particularly. And most of the very short answers, if you are seeing in the very short answers, we can't expect that actually this may be coming, but we have to see all the very short answers. We have to concentrate on the important points which we are seeing in the long answers and very short here. If you are covering the long answers and very short answers, in most of the long answers and very short answers, most of the very short answers will be covered. But particularly when we are writing the very short answers, remember that actually complete answer should be written. It may be 2 marks, but clear explanation of the very short answer should be given here, whatever the point may be. If you had about the penicillin antibiotic, who will get the credit of the penicillin antibiotic? We have to write about the Alexander Fleming, flora and chain. About all these things we have to write about the discovery of the penicillin and all these things. Then whatever the point you are writing, we have to write the point, we have to expand the point clearly, we have to explain the point clearly when you are writing about this thing. This is about the very short answers. We can't cover all the very short answers, but most of the very short answers we have been covered in this phenomenon.